Alright, hello everyone, let's get started. Good afternoon, it's already 12 o'clock. I get a chance to say good morning to those in the previous section. I can tell you good afternoon. So welcome to lecture 14 of the integumentary system. And as you will notice, I've said part 1 because my goal is to talk about the normal structure of the integumentary system. And then on Monday, when you come back, we will talk about what can go wrong. And I can show you some pretty gross pictures. Pretty gross, yeah. It won't be pretty. But we'll talk about things like cancer, moles, acne, and how we can take good care of our skin. So before we get started, I have a couple of review questions. So the dermis of the skin is an example of which of the following connective tissue? Is it an example of the areolar connective tissue, the adipose tissue, dense regular connective tissue, dense irregular? More than one of these answers is correct. Alright. Okay, most of you said more than one of the answers is correct. And you are right. The reason being that if you, we will talk about the dermis later in today's lecture. And there are two parts to the dermis. You have an upper layer which contains the areolar connective tissue and the lower layer which contains the dense irregular connective tissue. But then the right answer is not adipose tissue because the adipose tissue will be found below the dermis in the hypodermis, right? So that's something you want to keep in mind, and we'll talk about this later in today's lecture. Yesterday I told you that when you think about different types of connective tissue, you want to keep in mind what are the various cells found in these connective tissue. So this question is based on that. So chondrocytes are specialized cells that will be found in areolar connective tissue, dense connective tissue, fibroelastic cartilage, or bone. Which do you think is the right answer? Alright, the right answer is fibroelastic cartilage. So any type of cartilage, whether if it's fibroelastic, hyaline, or elastic cartilage, they would contain chondrocytes. And so chondrocytes is the right answer. Okay, now I have a question for you about the matrix. I told you keep in mind what are the cells and also what is the matrix surrounding those cells. And so the question is, proteoglycans refers to which of the following? Is it a large class of non-collagenous proteins, or is it proteins covalently linked to polysaccharides, or is it a clear matrix surrounding the fibers, or is it a large and highly charged polysaccharides? Okay, this is a plain recall question and it is right. So proteoglycans refers to the proteins which are covalently linked to the polysaccharides. And notice that it is these proteoglycans which will interact with water because the polysaccharides linked to them are highly charged, and so water will interact with it and have this gel-like matrix which will form. Okay, with that, we're going to begin with a quick overview of today's topic, which is the integumentary system. And I want to begin this overview by asking one question, which is how do the components of the integumentary system aid in its function? So there are two parts to this question. One is what is the function of the integumentary system, and the next thing is what are the components of the integumentary system. And so in today's lecture, I just discovered in the previous lecture that we won't have time to talk about the various functions of the integumentary system. So I will answer the part on what are the components of the integumentary system, and then on Monday we'll talk about the detailed functions, and when I promise there are a lot of functions. So when you consider the components of the integumentary system, you're going to start by looking at the three layers which you will observe. The first is the epidermis, and deeper to that you have the dermis, and then below that you have the hypodermis. And each of these layers will have different types of cells. And because the integumentary system is an organ system, it's going to have all the four types of the major tissues which you talked about. So you're going to see connective tissue, epithelial tissue, muscle tissue, and also nervous tissue. You're going to encounter all four as we talk about the various components of this integumentary system. And then you'll also see what are some of the appendages which are found in these layers. And examples of appendages include the hair follicles, or the sweat glands, sebaceous glands, and so on. One appendage which I will not discuss about in this lecture is the nails which you see at the tips of your skin. It's just made up of keratin, so I didn't think it was necessary to discuss that. Okay, so when you think about the integumentary system as a whole, it's interesting to note that that is the largest organ in your body. So it is going to cover 16% of your, it's going to entail 16% of your overall body weight. And because you have different appendages which are found in this integumentary system, this is known as your largest organ system as well. It's not just an organ, but it's also part of the largest organ system. And the skin is made up of two distinct layers. The outermost layer is the epidermis, and then below that, or deeper to that, you have the dermis. And deeper to the dermis, you have this hypodermis, also known as the superficial fascia. But technically, it's not part of the skin. And now what we're doing is we're going to start looking at what are the different parts of the epidermis. So the epidermis, it is described as being keratinized, stratified squamous epithelium. Right? And so I mentioned that it is keratinized because there will be a layer of keratin on the most superficial layer of the epidermis. And also notice that it is stratified. So when I say something is stratified, the first question you would ask me is, how many layers? You know, it's definitely got more than one layer. So how many layers? And the answer is, it depends. It depends on which type of skin you're looking at. Now we know that the skin on the soles of our feet and on the palms of our hands are quite different than the skin which we have hair growing out of. You know, so depending on the type of skin, you can have different layers. So if it's the thick skin, it's going to have 5 layers because there is extra layer known as stratum lucidum, which I've shown in the box here. Whereas if it's the thin skin, there will only be 4 other layers. So what are these 5 layers? You have stratum corneum, lucidum, granulosum, spinosum, and stratum basale. Now notice that each of these layers has the name stratum before the second name, because stratum just means layer. And I want to help you remember these different names by using a fun mnemonic. And the mnemonic is kind of ironic because it says, "Come, let's go sunbathing." And I say it is ironic because we will talk about the risks of increased exposure to sunlight. But I just think that it is ironic and funny, but it helps you remember the different layers of the epidermis. So we're going to start all the way with the bottommost layer, which is stratum basale, because the cells are going to be differentiating that layer, and then they're going to be pushed upwards. And as you notice here, the cells at stratum basale look very different than the ones which in the stratum corneum. So they're going to undergo changes and differences, and it makes more sense to start from the bottom and work our way up. Okay, so let's start with stratum basale. So you'll notice that this layer is also known as stratum germinativum because it is a single layer of cuboidal cells which are highly mitotic. So they are rapidly dividing, and they divide to give rise to 2 kinds of cells for our interest. One is mainly keratinocytes, and A smaller portion of the cells are melanocytes, which produce the pigment known as melanin. And we will see how the pigments produced by the melanocytes will enter the keratinocytes later in today's lecture. And we also see a couple of other cells. One is the tactile cells shown here as a Merkel cell. The exact function of a Merkel cell I will tell you when we discuss the different tactile receptors in the dermis. So we'll talk about all of that in one slide. So just hold off on that. And also these dendritic cells are known as Langerhans cells, and this cartoon does not show a dendritic cell. In fact, it shows a dendritic cell in one of the upper layers because that's where they're usually found, and we'll talk about its function then. And notice that this layer is going to be connected to the dermis by what are known as epidermal ridges. And those epidermal ridges are responsible for the different types of fingerprints we all have. And we will talk about how that happens and the physiological relevance of that also later in the presentation. So for now, we're just going to keep moving upward all the way to the surface. So the next layer is stratum spinosum, and keep in mind that each of these layers have a name like granulosum, spinosum, basale, because there's a different meaning behind why we give that name. So there has to be something spiny or prickly in this layer here. So let's see what that is. So we have around 8 to 10 layers of keratinocytes, and you'll notice that these keratinocytes are irregular or polyhedrali shaped. So they have this spinous processes sticking out of them. But it was found that this spinous process is basically an artifact which is left behind after the histological sites have been prepared. So if you take a closer look at these cells, you'll notice that the gap between these two keratinocytes, there are these lines connecting the two, right? So those are the spinous processes. And if you were to look at the same thing in an electron microscopic image, you see that these connections are seen between the cells. So that is the reason why they call this stratum spinosum. So here's my question for you. What do you think these connections between these cells are? I spent an entire lecture talking to you about how cells are connected and cell junctions. So you can turn to your neighbor or go back on your slides and go back to your slides. But can you tell me what type of connection would be found between these keratinocytes? Anybody with willing to answer?
I know the answer. You're correct.
So we talked about tight junctions, gap junctions, adhesions, anchoring junctions. Talk about all those things, but any break so what do you think connects them? Yes, all the way to the back.
I was thinking it looked like a tight junction, just because all the filaments in the electron microscope.
Okay, tight junctions is a good guess. Are there any other answers besides tight junctions? Yes.
Gap junction.
Gap junctions is also a good guess. Are there any other guesses? Yes.
Anchoring.
Anchoring junctions. Under anchoring junctions, we have different subdivisions. Which one specifically? This is great. You guys had your hands up, and few of you put your hands down. Which among the anchoring junctions? Fine. It's the desmosome. Right? Okay. But let's start with the tight junctions and work all the way down to see why we would not, why I would not say it's the other answers. So tight junctions, I told you, it's mainly going to be at the apical surface of the cells. But look at these; they're connecting all over, all around the cell. You see these junctions. So I would say it has to be a type of anchoring junction. And among the anchoring junctions, we see there are 4. 2 of those connect the cell to the extracellular matrix, right? Whereas the other 2 connect cells laterally. And among those 2, one is adherence junctions, the other one is the desmosomes. So it is desmosomes, which I mentioned, is found in tissue where there is which are subjected to greater mechanical stress, such as cardiac muscle or your skin. And so the answer is desmosomes. Right? So you see how you have to work through all of the options and be like, nope, it can't be this, can't be this, and then we come to desmosomes. All right, great. So I'm glad that we have an opportunity to practice what we learned 2 days ago as we add new material to our memory banks. So we see that there are other things to note in this layer, and that is there are many melanin granules which were produced by the melanocytes in the stratum basale. You see tons of melanin granules, and you will also notice that these Langerhans cells or dendritic cells are also abundant in this layer. So how exactly the Langerhans cells function, we will not talk about the details. I just will mention 2 things. One is that it is it's a cell which can engage in phagocytosis, but it can also act on antigens and further boost the immune response. The details of that makes more sense to discuss when you discuss the immune system. But I will remind you that I did briefly mention these dendritic cells when we talked about the tight junction barrier being affected by claudins not being dysregulated. I told you when claudins are dysregulated, you can have pathogens entering into the intestinal villi. That is detected by 2 cells. One is the mast cells, and also the other one, the macrophages, and by the dendritic cells. So these are the cells which can release cytokines to trigger the immune response. So I'll just mention that, but the details of the Langerhans cells we'll cover in the immune system. So moving up, we see that we come to the stratum granulosum, and yes, no surprises, you're going to find granules in these cells. And notice that there's only 3 to 5 rows of cells, and these cells are now flattened. When you start out in the stratum basale, you have cuboidal cells, but as they are differentiating and getting the melanin pigments and they're moving upward, they're starting to get flattened off, and that's why when they reach the apex, they become stratified squamous epithelium. And the reason for them flattening out is because these cells are gradually dying, and the reason they're dying is because they're getting farther and farther away from the blood supply, which will end at the dermis. So I told you the epithelial tissue is avascular, there's no blood vessels there, and so any sort of nutrients have to be passed on by diffusion. So the farther it gets away, these cells start dying. And also there is a process known as keratinization which takes place. So in this process, there's going to be a hard shell formed over these cells, but internally the organelles are also going to start degrading as the cells die. So all of this, these changes are taking place inside these cells in the stratum granulosum. So the question is, what are the granules, right? So the granules, you see there, the keratohyalin granules, and they contain a number of proteins which are beyond the scope of this lecture. But what I would point out is that this keratohyalin granules has a few functions which I do want you to keep in mind. Firstly, these granules cause the cells to be dehydrated. Secondly, they also cause a cross-linking between the keratin filaments. So keratin filaments were discussed in the earlier lectures when I was not here, and keratin is a type of intermediate filament, and the cross-linking is done between those filaments. And thirdly, it is these granules which are going to cause keratinization. So keratinization, as I mentioned, is going to be a hardening of the outer surface of the cell, and there are multiple proteins involved in this, but all you need to know is that the granules are involved in the hardening of this outer layer of cells, but you don't need to know what are the proteins involved in that. Okay, so with that, we continue moving upward and we come to stratum lucidum, which is found only in the thick skin. And notice that this contains 2 to 3 layers of flat or dead keratinocytes, and this layer appears clear for a couple of reasons. One, the organelles have all been degraded, but you also have this substance called eladin, which is a transformation product of keratohyalin. So that's why this layer looks very clear. And you will see this type of skin only in the soles of your hands and feet. So you may be wondering, okay, what is the thickness between a thick skin and a thin skin, right? And if you were to use everyday examples, thick skin would be as thick as a paper towel, whereas the thin skin would be as thin as a plastic sheet. So that would be the difference between their thickness. So we finally move all the way up to stratum corneum, which is the superficial, most superficial layer, and this is made up of 20 to 30 layers of dead keratinocytes, and they're filled with keratin, right? And we call this a horny layer because when you think about the horns of animals, they're also made up of keratin protein. Now notice that I say this 20 to 30 layers because depending on what kind of activity you're doing, you could have more layers than the others. For example, I have calluses on my hands here because I like to do pull-ups once in a while. And so if you do pull-ups or you like to play the guitar, then you will develop calluses, and so you can have more layers in your stratum corneum. And this is the thickest of all the 5 layers, right? And it accounts for almost 3/4 of the epidermal thickness, and you have glycoproteins which are secreted in this layer which make it water resistant. And there is also substances called defensins which are secreted, which help to have an immune response against microbes. So we see that the stratum corneum has a protective function which is not only physical because of the multiple layers, but it's also chemical because it's not allowing water to escape easily by this glycoprotein layer, and it's also offering a biological protection through the immune response which it can generate. Is that clear? So I've talked about the various components, but I'm also trying to highlight what are the functions of these various components in the functioning of your skin. So I have a question for all of you, right? So this type of design is known as henna design. So henna is a kind of pigment which you get from a plant, and it's not just in India, but multiple cultures in the world can use this kind of pigment to either color their beards, hair, or skin. And this pigment, it's once you apply it, it stays on for a couple of days and then starts wearing away, and by 2 to 3 weeks it's completely gone. So why do you think that is the case? So this is a think pair share question, so you can turn to your neighbors and talk and see if you can answer this question. Alright, do we have anyone else having the answer?
Yeah, I think I'm
Why does it wear off after some time? Yes?
stains the epidermis, which are layers of squamous cells that regenerate really fast.
Okay.
And so after the 1 to 3 weeks, your cells have regenerated, unlike a tattoo that goes deeper than the epidermis.
Okay, great. Yes, thank you. That's the right answer. So the answer is that the skin will regenerate and the upper layers will be sloughed off, and that's exactly right. And so we see that it takes 7 to 10 days for the cells from the stratum basale all the way to reach the stratum corneum. 7 to 10 days. And once it reaches that layer, it's going to stay there for a couple of weeks. And when you have the henna stain, it's going to go stain the upper layers of the cell, and it can hold on to that stain for a couple of weeks, because that's how long the stratum corneum stays on before it is going to be shed off. And this is also known as defoliation. And we know that within 25 to 45 days, the entire epidermis is going to be completely regenerated. And so that's why if you have a tan, you see that you're going to lose the tan after a couple of months, because your epidermis is being regenerated as well. You have a question in the back?
I was curious, like, why do certain inks stick more than others? Like, why does henna last for like 2 to 3 weeks when you draw yourself with Sharpie?
Yeah, it really depends on the kind of, great question. The question is, why do certain pigments tend to stain better than others? It really depends on their chemical composition and how they interact with the keratinocytes. So the answer is it depends. That seems to be the standard answer. We need to look at the chemical structure of that specific dye and how it interacts with keratin protein. So, yeah. Any other questions? Yeah. So, if there are no other questions, I just want to point out that this picture here, it looks like a bunch of shingles. This is actually electron micrograph picture of your stratum corneum, and because we shed skin so regularly across the entire lifetime, we'll shed around 40 pounds of skin, which is pretty cool. So now that we've discussed about the various layers of the epidermis, I'm going to just talk about the specific cells which are in the epidermis. And we talked about keratinocytes in quite a bit of detail. We know that it's the most abundant layer. I don't have anything more to add to that. Merkel cells, these are touch receptors, and I will add more to that in the upcoming slides, so just hold on, but we see that they're present and they are connected to free nerve endings. And Langerhans cells, I told you that we will discuss more about that in the immune system. But what I do want to talk about are melanocytes. So melanocytes, these are the cells which are located in the stratum basale, and they produce melanin. So that's the pigment which is responsible for our specific skin tones. And these pigments are going to be produced by the melanocytes in response to exposure to UVB radiation from the sunlight mostly. And these melanin pigments, they're going to be packaged in vesicles known as melanosomes, and these melanosomes are going to be passed on to the surrounding keratinocytes, right? And how these pigments are donated is through these long dendritic processes, right? And that's how these pigments are going to be transported. And rather than telling you how exactly it happens, I'm going to show you guys a fun video.
In this scene, a melanocyte develops two long dendrites, which introduce packages of pigment granules, or melanosomes, contained within vacuoles, into a flat keratinocyte. While the individual melanosomes are too small to be seen in the light microscope, the transferred vacuoles, containing many melanosomes, are visible. The melanosomes are left behind as the dendritic processes start to withdraw. The vacuoles open up inside the keratinocyte, dispersing pigment into the cytoplasm. Let's watch the pigment donation again.
Cool. All right. I hope you like that as much as I did. But the one thing which this video did not specify is that pigment donation is actually going to involve the keratinocytes phagocyting of these long processes from the melanocyte. So imagine that these long processes are coming from the keratinocyte and this keratinocyte from the melanocyte, and it's basically going to bite off the tips of those processes, and that's how it engulfs those melanosomes containing the melanin pigments. And once these pigments have entered the keratinocytes, these pigments are going to act as a shield for those the nucleus of those keratinocytes. And notice that these pigments, they're going to move towards the superficial aspect of the cell, and they can protect it from the UV radiations from the sun. So this picture here, it says dark and light, and I'm just wanting to point out that dark-skinned individuals will be producing more melanin. It's not that they have more melanocytes, they just are, the melanocytes they have produce more melanin. And this definitely makes sense because if you're living in a part of the world where you have intense exposure to the rays of the sun, then you need to have more melanin to protect you from the rays of the sun. So coming from a country like India, I live in the southern part, which is pretty close to the equator, and the people who are in that locality, they have much darker skin complexions. And you go all the way towards the north near Kashmir, you'll see people with much lighter skin complexion. So while we're talking about pigments, we see that there's a condition known as albinism, where you can end up with milky skin, flaxen hair, and even the iris in your eyes are light because the body lacks the melanin pigment. Now, a consequence of not having sufficient melanin is that you could increase your risk of cancer because you're exposing the skin to the rays of the sun. And we see that in some individuals, the reason why they can't produce melanin is because there's a defect in the enzyme which is involved in catalyzing that reaction to make melanin. Another condition is known as vitiligo, where the melanocytes just die in certain parts of the skin, or they're unable to produce melanin. And one of the reasons for this is that it's often an autoimmune disease where the body's immune system detects the melanocytes to be foreign and starts attacking it. So the way to treat it is that you could transplant melanocytes into the specific parts of the body where the melanin is not being produced. So it's not only these things which can be determined by looking at the skin tone, but there are other conditions we can also identify by seeing the skin tone. So do you see anything abnormal in this baby by looking at the skin tones? What color do you see? Do you see one of the ordinary? Yes? Blue, yeah, very good. It looks blue around the nose and mouth. So this is a condition known as cyanosis, and it's just referring to the bluish discoloration of the skin, and why there could be a couple of reasons why this happens. It's mainly linked with deoxygenation of the blood. So the blood is not able to sufficiently pick up oxygen from the lungs and pass it to the various parts of the body, because it's most likely a problem with the hemoglobin molecule not functioning normally. And so that's why it could lead to a blue discoloration. Yes?
I was curious, I read a study of this one guy who took too much of something called colloidal silver and turned himself blue.
Oh wow.
Is that, do you know anything?
I'm not sure about that study, but do send it, I'll look at it. So I'm not sure what the mechanism behind colloidal blue is, to turn himself blue. Yeah, that's fascinating. I might even use that in next year's class, but send me that article. Alright, so let's look at this person here. So what do you think? Did this person come back from vacation in Florida, or do you think something else may be the reason for this skin color? How many of you think he did go to Florida? No one thinks that, right? So this is a condition called bronze diabetes, and this is a condition where the body starts accumulating excess iron because there is a mutation in the gene which will help absorb iron, and so there's no control and the body starts absorbing excessive iron. And this is a condition known as hemochromatosis. And when you think about iron absorption, there is no way for the body to actually excrete the iron once it is entered the cell. So the way the body functions normally is that when you have iron in the food, it enters your small intestines, it enters the intestinal cells, the enterocytes, and it's going to stay there, and the body will shut down the mechanisms to make the iron exit from the enterocyte into the bloodstream. And so the iron stays in the enterocytes. Within a couple of days, the cells get regenerated and the iron gets sloughed off. Right? So that's how the body controls it. But if the genes which control the iron entering into the body is not functioning, then it will start accumulating excess iron, and it can eventually lead to a condition like this. I don't expect you to know the mechanisms of how this happens, just know that excess iron accumulation can lead to the skin bronzing. Okay, what do you think this condition is for these gentlemen? Jaundice, yes. And so the reason for the yellowing of the whites of the eye and also the mucous membranes turning yellow is because of a pigment called bilirubin. So bilirubin is the main pigment in bile, but the way bilirubin is produced is that it's a breakdown product of the red blood cells. And we can talk about these details in the upcoming lectures. But the reason why this bilirubin is not excreted normally could be for a number of reasons. Either the liver, where the bile is being produced, may have some problems because of hepatitis, or the bladder from which the bile should be secreted out may be blocked because of gallstones, which are blocking those holes, blocking the openings, and also tumors, which could cause dysregulation of how bile is being produced. Okay, what about this little guy here? Any guesses?
It's from an excess amount of carotene.
Exactly, perfect. It is called keratonemia, and this can happen if you have too much foods containing high levels of carotene, like either juice or juice from fruits or vegetables. And so sometimes it could be thought that this condition is jaundice and can lead to an unnecessary investigation, but this is quite harmless. And if you remember what I mentioned yesterday about adipose tissue, keratin is fat soluble, and so if you have it in excess, you just can't pee it out. It's going to be stored in your fat cells, and that's why the skin starts turning yellow as well. So now we're going to move from being just skin deep and talking about color to talk a little more about the dermis. We're going to move down to the lower layer of the integumentary. So in the dermis, there are two layers. The first layer is the papillary layer, and this only contributes to 20% of the thickness of the dermis. And it's mainly consisting of areolar connective tissue. And notice here that this is the areolar connective tissue. It has elastic fibers and fibroblasts, but notice that they're arranged pretty loosely. Whereas in the reticular layer, it is going to be the remaining 80% of the dermis, and it has dense, irregular connective tissue. That's the key here. It's not a dense, irregular connective tissue, but it's dense, irregular connective tissue, and it has these abundance of collagen fibers and the elastic fibers, which allows both strength and the residency of the skin. So, any questions at that point? Okay, moving on. We're going to talk a little more about the papillary layer of the dermis. So you notice that this papillary layer has got these peg-like projections called dermal papillae, and it is these peg-like projections which help attach to the epidermis. Right? And so we will see how these projections interact with the epidermis to lead to the epidermal ridges, which are eventually responsible for the fingerprints. But I want to point out a couple of things at this interface here. There are also these capillary loops which contain, which are blood vessels, and that is how nutrients can reach from the dermis into the epidermis, especially in the stratum basale. There are also these Meissner's corpuscles, which are tactile receptors, and I'll talk about the functions of tactile receptors altogether. And you will notice that there are also free nerve endings, which are acting as pain receptors as well. So if you look at this picture here, you have these epidermal ridges, which are the raised area, and these darker lines are the papillary ridges, and these interact together to attach the epidermis to the dermis. Now, the way in which these epidermal ridges are formed are going to be unique between individuals, and while we use it for fingerprinting and identifying people, the physiological function of this is to increase the surface area of the skin and to improve the ability to grip things. And this will start developing in utero as early as 10 weeks. The fetus is only 80 millimeters long. So another thing to keep in mind when we talk about these epidermal ridges are the cleavage or tension lines seen throughout the skin. So these lines, they are formed by the arrangement of collagen and elastic fibers in parallel bundles. And because they are arranged in a certain orientation, they're going to resist forces to stretch them in a specific direction, right? And there's a pretty specific clinical implication of this, and I'll point that out to you. You have a question?
I was just curious, is this why stretch marks form when you do?
Yes. So the clinical implication for this is going to be that if you have an injury, if that cut is made parallel to these cleavage or tension lines, then it's going to heal without a scar. But if it's going to be made perpendicular to these lines, then it will form a scar. So if you think about specific types of surgeries happen, you'll notice that the cuts they make will be alongside these cleavage or tension lines. So the question asked was about why do stretch marks occur? Stretch marks occur because of the skin is in a certain direction, and if it starts stretching, then very rapidly, then it doesn't have sufficient time to accommodate those changes in shape, and so it could lead to stretch marks. So now what we're going to do, we're going to just quickly talk about the various components of the dermis, and I'll briefly point out what their functions are. So we see that there are blood vessels which bring our nutrients and to the skin, and also take away waste products. And here's the hair follicle, which can detect changes in the environment because it's connected to nervous tissue at its base. There are sebaceous glands which secrete oily substances which will not only help lubricate the skin and to help retain moisture, but it also has antimicrobial activity. And sweat glands, when you secrete sweat, it not only helps with thermoregulation by cooling your body down, but it can also help with excretory function to get rid of waste products. And last but not least, notice that there is this erector pili muscle which is connected to the hair follicle. So this is where we encounter muscular tissue in the integumentary system. So this muscle is not under our voluntary control, and it's under involuntary control. And if you're scared or cold, when this muscle contracts, it's going to cause this hair to stand up straight, and it leads to what is known as goosebumps. So now let's talk about the different types of sensory receptors. We start with the Merkel cells, which are the only type of tactile receptors in the epidermis, and this can detect sustained pressure. So if you were to press your skin, you can, for a few seconds, then it's detected by your Merkel cells, because they take a while to adapt to the external environment, and so it can be detected by it can detect those slow pressure which is applied to your skin. On the other hand, you have these Meissner's corpuscles, which are found in the dermis, so in the layer below, and it can also detect light pressure as well. But notice that the difference is they are in different parts of the integumentary system. So if you come deeper, you have the Ruffini corpuscles and Pacinian corpuscles, and notice that the slides I posted, Ruffini corpuscles have a different function, so make sure you note this one down. The Ruffini corpuscles and Pacinian corpuscles, though they are located deep in the dermis, they have different functions. So these corpuscles can detect stretches, and they can also, if there's any sort of change in temperature, that can be detected by these corpuscles. And Pacinian corpuscles, they can detect deep pressure, and they can also adapt pretty quickly to this deep pressure and can be sensed by the body. And finally, notice that the reason why if a mosquito were to sit on your hand, and it can distort the hair, you can feel it because of these root hair plexus, which are also nerve endings found at the base of the hair follicle, and that can also send a signal to the brain. So with that, we conclude our discussion on the dermis, and we go all the way down to the hypodermis. So the hypodermis, you notice that it consists mostly of adipose tissue, and its function is to absorb shock. It also can act as an insulator to reduce the heat loss, and it helps to store energy reserves. And this is the deepest layer of the skin, and it's not really part of the skin. It's called the superficial fascia. And notice that this hypodermis is mainly composed of the areolar connective tissue, and it's anchored to the muscles. And deeper to this, you will have subcutaneous fat, which is where you will mostly store fat. But in some cases, if you exceed this subcutaneous fat capacity, then you can start storing fat in other parts of your body as well. So I have a couple of questions for you. So what layer of the skin do you think dye is injected into for when you put a tattoo? Is it the epidermis, dermis, hypodermis, or the underlying connective adipose tissue? Oh, oops, sorry. So most of you said dermis, and that is the right answer. And so that's great. So if it wasn't the epidermis, it would get sloughed off like the henna dye. If you put it in the hypodermis, you can't see it, right? So you want to be able to see it. So it has to be the dermis. Okay, another question. So leather is produced from what component of the integumentary system? Is it epidermis, dermis, or hypodermis? Oh, most of you think it's the epidermis, but the right answer is the dermis, because that's where you have all of that connective tissue, especially that dense, irregular connective tissue, which helps to give the leather belt. So I'm going to quickly summarize what we've discussed. So my goal was to talk about the components of the integumentary system, and we talked about the 3 layers of the integumentary system. We talked about how the epidermis has got 4 to 5 layers. It has a protective function. We talked about the dermis, which has 2 layers, and it has 2 types of connective tissue in those layers. The hypodermis has mainly an insulatory function, and also it stores fat. And we discussed about the various types of cells, and specifically we focused on the keratinocytes, which have a protective function, and melanocytes, which also has a protective function by secreting melanin directly from UV radiations. In terms of sensory receptors, we talked about different types of sensory receptors, so I would expect you to know what their functions are and where they are found. And we see that the integumentary system is associated with all 4 types of tissues: connective, epithelial, muscular, and the nervous tissue. And the appendages we discussed included sweat glands, the hair follicle, sebaceous glands, blood vessels, and also the adductor villi muscles. So now that we've discussed about all of these components, we will see how they help to help the function of the integumentary system. And before you leave, I have one piece of news for you. By 5 p.m. today, we will have the results ready. And I want you to know that I've never seen a more beautiful normal distribution, right? So it means I did my job in communicating the information to you. But the good news is that you get to drop one exam, right? And I really want you to make sure that if you got any question wrong, try to find out where the answer is. And if you can't find it, come and talk to us, because I really hope this is a stepping stone for doing better. Because the material going forward will get more complex because you're going to add more material together, and I want you all to do well. Enjoy your weekend.
Is it just going to be like the Z-strip thing and like your answers and the other answers, or is it just going to be this is the key? And
I believe they're going to give you back your answers.
Okay.
Yeah, because I want students to be able to
Let me just get the
let me talk to you. I really feel that exams are a fantastic tool for you guys to learn, you know? Because there's nothing better motivating people to sit and pay attention. And I'll also make sure that we have a review session on the day of the exam in class so that you all can do it.
Sounds good. Thank you.
Alright, everybody, enjoy your weekend.
You as well.